of the Nodia district of West Bengal. This is the collection site 1, and this is the site of the Morali River. Actually, Morali River, it is the rivers of the Nodia district. It is also called a lost river of the West Bengal, as due to the high due to the high accumulation of the organic matter, it become the wetlands. So this collection site 1 is very much associated with the human habitats and human effluent. We have collected the Paila sp and Belamnia bengalensis from this aquatic site where the human interference and anthropogenic source occur. Next, collection site 2. This site has not been interfered by any type of the human activity. This is the site 2 of the Morali River. That is very much near to the Morali Bridge, Silinda, Daragpur or Horinanka of the Nodia district. The water is completely devoid of any type of the pollution, because there is no human habitat and there is no chance of the accumulation of the human effluent. We have collected the specimen from there, and this is the picture of the collection 2 site, and this is the picture of collection 2 of the Horiyanka of Nodia district. Analysis of the hemocyte and cells from the hepatopancreas. Now the left hand side upper portion, we have collected the hemolymph. Hemolymph is the blood fluid of the mollusca, and stained the hemocyte with the help of the Giemsa and methylene blue. And in the right hand side, we found the accumulation of the hemocyte. It is called as the aggregation of the hemocyte of the Belamnia bengalensis. And the lower portion, that is the habitat that is completely interfered with the human effluent and pollution. There is no aggregation. All the cells are very much individual. The human effluent is very much associated. This is the neutral red positive response. In the right hand side, the cell, only the lysosome took the neutral red color. Only the lysosome took the neutral red color, because the site of the selection, it is not polluted. But the right hand side, not only the lysosome, the cytoplasm also is the neutral red positive response. It means the lysosomal membrane is broken. As a result, lysosomal enzyme come into the cytoplasm. As a result, the neutral red took color in both lysosome and the cytoplasm. As we have collected the specimen from the site where the human habitat or human discharge occur, the cells are damaged, lysosomal enzyme come into the cytoplasm, and that is called as neutral red retention assay. This is called as the trypan blue assay. Trypan blue means, trypan blue is the stain that only give the positive response to the dead cell. If the cell is dead, it will react to the trypan blue and give the blue color. The left hand side, the specimen collected from the polluted side, so all the cells are blue in color, and the right hand cell, the very few cells are blue in color. The cells that are white in color, that too did not take the trypan blue coloration. Mean number of trypan blue positive cells in the Belamnia, that is the mollusca, is high in the, it means the area that is very much associated with the that is very much associated with the mean number of the is high in the polluted area, and the p value is 0.0018, that is also statistically significant. This is the collection site 3. Actually this region is very much associated with the agricultural field, that is Raghunathpur. It is associated with the Nakasipara area of the Nodia district, where the this aquatic area is associated with the agricultural field where the pesticides and insecticides are used regularly. Now from this area we have collected the Pila and Belamnia sp. So study of the molluscan hepatopancreas. Why we have studied pancreas? Because the hepatopancreas, it is the detoxifying structure. It is very much similar to the liver of us. Mammalian liver equal to the molluscan hepatopancreas. These hepatopancreas contain different types of the cells like digestive cells, basophilic cell, duct cell, embryonic stem cell, etc. Now this is the pila and this is the hepatopancreas. We have dissected out the hepatopancreas. We have mixed the tissue with the trypsin EDTA, and the cell suspension is used for the trypan blue solution and stained with the Giemsa. The left hand side, it is the cells of the hepatopancreas from the non-polluted area where you can find the distinct nucleus. But in case of the right side cells, the cell membrane degenerated, nucleus degenerated, apoptosis like behavior occur, inner cytoplasm has been released outside the cell. It is also similar to the necrosis. Mean number of the pycnotic cell from the hepatopancreas of the pila is very much high in the polluted area, the area that is related to the agricultural field. I think this is due to the use of the insecticide and pesticide. And the p value is 0.0015. It is also statistically significant. And this is the hepatopancreatic cell in the of the Belamnia bengalensis. It is in the left hand side, the control cell. Right hand side, it is the necrotic cell or apoptotic cell. You can find the rupture of the cell also due to the use of the insecticide or pesticide. Mean number of the pycnotic cells from the hepatopancreas, it is very much high in the B, that is the pollutant and the p value is 0.0034. It is also statistically significant. Now the hepatopancreatic cell react with the trypan blue. The left hand side, it is the live cell from the non-polluted area, so they did not give the trypan blue coloration. But in case of the right side cell, most of the cells become trypan blue positive because we have collected these cells, this specimen from the polluted area due to the interference of the insecticide and pesticide. And the mean number of the trypan blue positive cells of the hepatopancreas in case of the pila is high. In case of the polluted area, the p value is 0.0002, that is also less than 0.05. So it is statistically significant also. And this is also the neutral red coloration due to the lysosomal damage. Both lysosome and cytoplasm become red in color. But in case of the control, because where there is no pollution, no human interference, these cells only the lysosome contain the red color. Upper portion only the lysosome contain red color. The lower portion picture both lysosome and cytoplasm contain red color. It means lysosomal breakdown or the cellular death. So what is the discussion? The discussion is that the specimen that we have collected from the human habitats and human effluents has been associated and related to the agricultural field due to use of insecticide and pesticide. They may hamper the environmental condition and also the freshwater ecosystem. Now, why we have collected the hepatopancreas? Because due to the high use of the human excreta or anthropogenic activities, we know that algal bloom occur. One of the important algae is cyanobacteria. Cyanobacteria produce cytotoxin, and one of the important cytotoxin that can hamper the liver called hepatotoxic microtoxin. So the liver of the mollusca is called as the hepatopancreas. That is the site of the detoxification. And there are many work from the other scientists that the agricultural runoff, pesticide and insecticide have been hampered the hemocyte function and cells of the hepatopancreas of different type of the mollusca of the animal kingdom. So what is the conclusion? The environmental challenges may alter the morphology and behavior of the hemocyte and hepatopancreas of the mollusca, but comprehensive research is necessary to identify the bioindicator to address the environmental challenges. This is the references or literature cited I have consulted. Thank you very much. And again, thanks the organizing committee for selection of my abstract and select me to provide the oral presentation in the online mode. Thank you very much.
Thank you, Shukanto, for your excellent presentation. Now I request Shaki Tamang. I think he is not here. Dr. Shantanu Chatterjee. Hello.
Yes, yes, sir. I'm here.
Please start your lecture. Yes. And I am requesting you all, please complete your lecture within 10 minutes. Okay. Okay. Thank you, Shukanto, again. Shantanu, madam.
Yes, sir. Sir, my screen is visible.
Yes, you are audible as well as it's complete.
Okay.
Please start.
Good afternoon, everyone present here. I am Dr. Shantanu Chatterjee, faculty member, Chemistry Department, Vijayagarh Jyotish Rai College. Today, I am going to discuss about hydroxy-9,10 anthraquinones and its metal complexes and their resemblance with anti-cancer drugs, for example, anthracyclines. In this context, as we know that cancer has become an epidemic in our society today, and a recent survey also showed that cancer has touched every family at least once and is now a serious cause of concern. So far, its treatment is concerned, chemotherapy and radiotherapy in conjunction with surgery has proved to be the most significant approach in this regard. One very important chemotherapeutic drug is anthracycline and its analogs. Anthracyclines actually show their anti-cancer activity by forming an intercalation complex with DNA and thereby inhibit DNA replication and RNA transcription. But the significant problem with this anthracycline is their high cost and cardiotoxic side effects. Hence, efforts have been made to find out mimic molecules. In this regard, we have chosen for our study hydroxy-9,10 anthraquinone. This is the structure, and we can see that this is the core moiety, and which can mimic the core moiety of anthracycline drugs, that is hydroxy-9,10 anthraquinone, which is mimicking these drugs. And we have chosen 1258 tetrahydroxy-9,10 anthraquinone. Now the question is, why quinazilazine is chosen? It has similar binding sites as that of anthracyclines. It also inhibits DNA topoisomerase 1 and 2 enzymes and reduce generation of free radicals. Additionally, its metal complexes are DMSO and water soluble. And it is cost effective, and one additional quality of this ligand is that it is casein kinase 2 inhibit. It can inhibit casein kinase 2, which is responsible for cell growths. So we have chosen quinalizarine, and we have taken quinalizarine and recrystallized from an alcohol water mixture, and we have taken metal complex as copper nitrate and carthamus DNA from sigma. Now the methods applied were the physicochemical methods. First, we are talking about the physicochemical methods. For stoichiometry determination, mole ratio method and Jobs methods were applied, and both the methods indicate that 1:2 is the stoichiometry of THAQ in presence of copper. Then the determination of PK values of queen alizarin using spectrophotometric titration were done, where two PK curve we have obtained, and in case of in presence of copper ion, the ligand was showing one PK curve, which confirms the complex formation. And here is a comparison of PK values of hydroxy 910 anthraquinone with anthracyclines, and it is showing close resemblance with that. And this is our compound. Now, the formation constant of copper complex of THAQ were also was in a good agreement with that of copper adiamycin complex, we can see from this table. And the absorption spectra of THAQ, that is the queen alizarin, in presence of at pH 7.56, in absence and in presence of copper ion was taken, and it was showing that in presence of copper ion it was giving a red shift, a prominent red shift. Then the complex was prepared by refluxing the copper nitrate and THAQ and by mixing it with 1:2 ratio. And it was analyzed by CHN analysis, IR, mass, EPR. In IR, the sharp peak obtained, one sharp peak obtained for phenolic OH at 3379 centimeter inverse. But in case of the metal complex, the peak is broadened due to the, which indicates that the OH group on THAQ was involved in the binding with copper. Then mass spectra was also obtained, and then the binding of THAQ to capthymus DNA at different pH was done, and the double reciprocal plots, non-linear plots were maintained to form the apparent dissociation constant and then the overall dissociation constant. And to understand the role of the metal complex or the ligand as anti-cancer agent, it is important to know how it is reacting with DNA. It was followed under UV visible spectroscopy and fluorescence spectroscopy for the metal complex. The binding parameters obtained for THAQ, that is the ligand, queen alizarin, interacting with DNA indicates that overall binding constant is almost 10 to 40 times less than that of reported anthracyclines, probably due to the absence of sugar moiety. But in case of the THAQ, it is less costly, and the superoxide formation is much lower than that of the anthracyclines, which is mainly responsible for the cytotoxicity as well as cardiotoxicity. Now, in case of the copper complex, we have got the comparable DNA binding values with that of the ligand. Now, the enzymatic assay was performed, and to calculate the superoxide formation. And superoxide formation of THAQ, that is queen alizarin, and its copper complexes were determined spectrophotometrically by the rate of superoxide dismutase inhibited cytochrome C reduction. And it was found that in case of metal complex, a carbonyl of each queen alizarin is engaged to copper ion. Hence, the probability of getting superoxide when semiquinone is oxidized by molecular oxygen is decreases. So, the formation of the free radical also decreases in case of which we can see clearly that in case of metal complex, the superoxide formation is significantly decreases. Now, THAQ, we have done the topoisomerase inhibition studies. The queen alizarin do not show any sign of inhibition, but through complex formation, this is able to inhibit the DNA topoisomerase 1 and 2, as the ladders are absent here, and here the ladders are present. So, the DNA topoisomerase inhibition is working in case of complex only, in case of queen alizarin. Now, we have done an MTT assay was performed using THAQ and metal complex of quinalizarin on acute lymphoblastic leukemia ALL mold 4 cells, and results suggest that THAQ is better performing than its copper complex. And as we can see that this is THAQ, and the blue one is THAQ, and the black one is copper THAQ, copper complex. And this is the another ligand, that is red one, that is not discussed in this study. So this is a summary of the findings, and from this we can say that that it is generally accepted that anthracyclines are cytotoxic to cells owing to their ability to generate superoxide radicals. From that point, quinalizanin is quite effective in generating superoxide radical anions, and the complex is not like that. And thereby, in case of complex, cardiotoxicity is lesser. But these findings are also indicating to the mechanism of anti-cancer activity of quinalizanin and this metal complex. In case of metal complex, both the complex metal ligand and the metal complex are cytotoxic, somewhat lower than anthracyclines, but they are cytotoxic. But the cytotoxicity of the ligand THAQ or quinalizanin is due to their superoxide radical generation. But in case of metal complex, it is doing the cytotoxicity or anti-cancer activity by topoisomerase inhibition. And here we can conclude by some summarize the fact that hydroxy 910 anthraquinones are much less costly, and it has similar binding sites as that of anthracyclines, and it is soluble in water and may be used under physiological conditions. DNA binding is slightly lower, but in case of metal complex formation, it can be increased. But in our case, the copper quinalizarin complex shows comparable DNA binding ability to quinalizarin. It is a better inhibitor of human DNA topoisomerase 1 and 2 than quinalizarin. The complex produces less semiquinone and superoxide radical anions, and lower radical generation likely reduces cytotoxicity in MALT4 cells despite strong topoisomerase inhibition. The study indicates the free radical pathway plays a dominant role in cytotoxicity for the ligand, and the topoisomerase inhibition might be the pathway of cytotoxicity of the metal complex, and it is showing lesser free radical generation, so the cardiotoxicity might be lesser. So these are the references I have used, and I am grateful to Professor Saurabh Dash, Jadavpur University, Chemistry Department. I am thankful to Professor Hemanta Kumar Mojumdar, IICB faculty member. I am thankful to Chetan Kumar, research scholar, IICB. And I am thankful to the organizers who have given me the chance to present my presentation. And I am also thankful to our principal madam and all of my colleagues for their help and cooperation. Thank you for your patience.
Thank you, madam, for your wonderful lecture. Okay, now I request Dr. Ratna Sarkar for her presentation. Ratna madam, please start. Hello, madam. Are you ready for the presentation? Ratna Sarkar. হ্যাঁ উনি আছেন দেখছি হ্যালো হ্যালো ম্যাডাম আচ্ছা তাহলে তারপরে দেবুন কি ডক্টর শুভঙ্কর সর্দার আচ্ছা প্লিজ স্টার্ট ইউর oral presentation, okay? Then we'll proceed, okay? Please, Shubhankar Sardar.
Is my slide is visible, sir?
You are audible and it's visible. Please start. Okay.
I am Dr. Shubhankar Sardar, Department of College Dato, So, my lecture topic is 1,3-diphenyl isobenzofuran aryl silane derivatives for improved organic transistor application and photophysical characteristics. Actually, this is a what? So there is a large number of paper and many discussion in my paper, but I am not presenting those in this oral presentation. I presented here only the conclusions in this oral presentation. And first of all, I would like to thank the all organizers for selecting my PPT for the oral presentation in this international seminar. So, first of all, what we have done, we have investigated some aryl silane based on the 1,3-diphenyl isobenzofuran as potential new organic semiconductor candidate to determine their bands, gaps, and reactance energies, both electron reorganization energies of the aryl silanes and the reactive calculations are performed at the 3 Kelvin versus 6113 cluster level. The results reveal the three suggested aryl silanes have superior properties compared to the other semiconductor materials. Geometry and electronic structure and the absorption or emission spectra of these 14 molecules are calculated at E0, EF2, triple zeta, EP level. Ground and intermediate compounds of aryl in gas phase and different organic solvents are studied. Compounds which have reorganization, whole reorganization energy is greater than the electron reorganization energy are better electron transport material or ETMs, and those with the reorganization energy whole is less than the reorganization energy electron are better whole transport material. And further, the study shows that this molecule can be employed as effective trifunctional material, that is the emitter, hole, and electron transporters in the OLED due to their improved electron injection and transport balance. We have presented this work in my two papers. One is published in 2024 and one this year in Computational and Theoretical Chemistry Journal. And these are the molecule we have studied in the gas phase and in different solvents. These are the structure of the molecules. And I have named them A0, A1, A2, and like that. So what we have studied 12 such model molecules, and we have studied their properties. Here you can see the HOMO and LUMO energy level, and this A1 is our starting material, and after the substitution with several electron donating and withdrawing groups, we obtain these compounds AA0 to AA11, total 12 compounds. Here we have find out different properties like the ionization potential, hole extraction potential, electron affinity, electron extension potential, reorganization energies, electronegativities. All quantities are in electron volt. We have compared their lambda H values, and we see that lambda 5 is less than lambda a when we are comparing the lambda H values, what we have seen, AA5 is less than AA2, is less than AA1, is less than AA3, and less than AA4 and AA0. In this connection, 4-methyl groups makes the dimeric DPBS less conjugated and unstable. Therefore, the 10 becomes AA0 is less than AA3 is less than S1. Isomeric AA4 and AA5 shows ionization potential value 5.57, 2 and 5.61 electron volt respectively due to good conjugation and the easy availability of the electrons. Unlike IP values, these molecules show different trends in the electron affinity values as follows. AA1 is greater than AA4, is greater than AA5, is greater than AA0, and the least value is shown by the S1, our starting material. Here is a table 2. This is the some theoretical spectroscopic data. The electronic transition energies are in electron volt, oscillatory intensity of the absorption, exciton binding energies, absorption and the emission maxima of the molecule under study. Uh, that is A1, S1 and A0 to A11. This table shows that the Array's length shows the excitation in a singlet state S0 to S1 state, and all electronic excitation are from the pi to pi star transition. These transitions are only associated with the electrons being promoted mostly by from HOMO to LUMO, and some fewer transition we have observed were HOMO -1 to LUMO and HOMO to LUMO +1. The experimental absorption and the emission spectra of DPBF are found to be lambda max value 411 nanometer. These values are in reasonable agreement with the lambda max calculated at the 459.7 nanometer and 4.13.1 nanometer. What we have seen, the 90% transition, what we have observed from the HOMO to LUMO, and they are from the pi to pi star transitions. And we came to the summary section. As possible candidate for new organic semiconductors and singlet fission material, we have theoretically investigated the photophysical characteristic and the optoelectronic uses of the 12 aryl silanes based on the defined connected dimeric DPBS molecules. By incorporating defined connections inside the parent structure of two DPBS, the hold and the electron transport characteristics have been modified for the aryl silanes. Our predicted molecules with band structures, band gap, density of states, excitation state energies, absorption and the emission wavelengths, and other factors are in good agreement with our previous experimental findings and data from other well-known OLED materials. Several contradictory requirements like the low HOMO, low ionization potential, and the whole extraction potential, lambda H is less than lambda E, reasonable force constant, and so many parameters we have applied in order to select the best whole transport materials. It has been concluded that A1, A2, and A3 are appropriate whole transport materials features after accounting all of these factors, and A7 can be used as an ambipolar material because of their slight lambda H and the lambda E difference. Upon photophysical characterization of the DPBS derivatives, it is demonstrated that the primary source of absorption and the emission correspond to S0 to S1 transition and that of pi to pi star character and HOMO to LUMO mainly. Among all the molecules that have been investigated, AA1, AA2, and AA3 are theoretically projected to be the best singlet fission material with good open circuit voltage, fill factor, and power conversion efficiency. Thank you for your patience, and thank you for giving me opportunity to give my oral presentation here. Thank you, sir.
And thank you, Shubhankar, for your excellent presentation. Now I request Ratna Sarkar. Hello, Ratna. Ratna
Yes,
Sarkar.
sir.
Please start your presentation, okay?
Okay.
Please, madam, please start.
Okay. Thank you to all. Very good afternoon. Today my topic is an eco-friendly method of synthesizing bismuth oxide iodide nanocrystal for wastewater remediation. And This is the outline of this presentation. Objectives, introduction, synthesis procedure, detailed characterization, degradation of rhodamine B dye, possible mechanism, conclusion and acknowledgement. Objectives. Initially, we try to develop the suitable synthesis strategy for bismuth oxyiodide nanocrystals with different morphologies. Then to characterize the material by different sophisticated techniques for detailed insight into its property. To examine the as synthesized material as a catalyst for photocatalytic degradation of rhodamine by rhodamine B dye in water environment, and then to examine the effect of different controlling parameter on the catalytic activity of the material. Finally, to propose a possible mechanism for the catalytic reduction of rhodamine B dye by the as prepared material. Introduction. In chemistry, photocatalysis is the acceleration of a photoreaction in the presence of a catalyst. Rhodamine B dye is a dark green color organic dye. It is wide used in industrial purpose such as printing, dyeing, textile, paper, paint, and leathers, plastics, etc. Various industrial solvents. Its release in the environment causes severe detrimental effect on the aquatic ecosystem. This is the synthesis procedure. We use bismuth oxide as a precaution material, and Ki is a salt, and PPP used as a capping agent. And all sample is diluted by DI water. Then it place probe sonication for certain time. After that, the material is washed and dried. And here we prepare two material. One is PVP added material, and other is no PVP added material. Now come to the interesting results. This is first figure shows the XRD structure of the both material. Here the all characteristics peaks are available for the PVP added or no added material. Then the figure B shows the FTIR spectrum for both material, and there is also all characteristics peaks are available, and the both from the both figure we conclude we see the material is proper formation. This is the first figure is the DRS reflectance spectroscopy, and the inside of the figure A is the calculated band gap from the Kubelkamp function, and the band gap of the both material is lies in visible range, and figure B shows the PL photoluminescence spectra for both samples. The no PPP sample is high intensity than the added PPP samples. Now come to the morphological analysis. First figure is the no PPP sample, and the second figure is with PPP sample. And all materials are nanomaterials. Now come to the catalytic activity results. First figure is absorbance spectra for no PPP material, and second one is with PPP material. And this is the catalytic analysis. Figure A shows the CT by C0 versus the time, and figure B shows the ln C0 CT versus time. And from this figure, we calculated the linear fitting for both samples, and from a calculation we get the weight constant for both samples. With PPP samples have high rate constant for mother. And figure D is the degradation efficiency. for both bar plot for both samples with PPP materials is highly decreased efficiency from others. This is the various catalytic um, performance. Uh, First one is dosage variation. Second one is stability test. Third one is pH variation, and number D is the scavenger test. Here we show the EDTA is a highly effective material, and this is whole quencher scavenger. And this is the possible mechanism. And here we use rhodamine B dye as a pollutant, and the whole mechanism is occurred in a visible light region, and the whole is the highly active material than superoxide anion. Now, final come to the conclusion. We were successfully in the development of a PPP varied pure bismuth oxide nanomaterials and bulk material by hydrothermal synthesis uh, uh, process, and characterizing techniques we use XRD, FTIR, UVV DRS, PL, and morphological analysis we use FSM, and the as synthesis material we see the catalytic property within 100 minutes, and then optimize the sample for 30 mg, and finally we propose a possible mechanism for the degradation process. And now acknowledgement. I acknowledge my college, and I acknowledge my supervisors, and I also thank you, the organizing committee, for the seminar. And finally, I thank my lab mates, family member. Thank you for patience. Any question? Hello?
Hello. Thank you, ma'am, for your excellent presentation. Now I request Shakti Tamang for his.
No, any question? Okay, thank you.
Okay, thank you, ma'am. Now I request Shakti Tamang, okay, Botany Department Research Scholar, for her presentation. Okay, Shakti, please start.
You want to present? Come by.
Yes, sir.
Okay. अच्छे से करो. Yes, sir.
Sir.
Shakti, please share your screen, okay?
Yes, sir.
Yes.
Sir,
now,
is it visible?
Yes, yes. Now it is visible. Please start.
Good afternoon, everyone. Okay. I'm Sakshit Tamang, research scholar at Department of Botany, Darjeeling Government College, and today I'll be presenting my work on comparative metabolomics of flavor-associated metabolites in TS569 and P312 Chinese tea varieties under the supervision of Dr. Saurabh Chakrabarty at the Plant Biochemistry and Molecular Biology Laboratory, Darjeeling Government College. Okay, introduction. Okay, tea, scientific name Camellia sinensis, is one of the most popular beverages in the world, valued for its unique flavor, aroma, and health benefits. These characteristics are largely influenced by secondary metabolites, specialized plant compounds such as terpenes, catechins, flavonoids, and theanins. Advances in techniques like GCMS and NMR have allowed us to identify over 700 volatile aroma in tea. In this study, we evaluated TS569, a seed-grown variety with high yield potential, selectively bred under strict standard P312, a garden clone of Chinese origin known for its aromatic quality. The main focus of this study was to analyze and compare the flavor-associated metabolites profile of these two varieties. Okay, objectives. The objectives of this work were to identify major metabolites contributing to flavor in TS569 and P312 and to compare the metabolic composition between both varieties, to study biosynthetic pathway for bioactive compounds. Materials and method. Fresh leaves from both varieties were collected from Darjeeling tea estates. Samples were washed, dried, powdered, and extracted in HPLC grade methanol for 48 hours. And the extract was, and then the GCMS analysis was then performed to profile metabolites. For pathway studies, we use bioinformatics tools like Cake Pathway Database and PubChem. Result: in TS569, we detected 79 compounds on GCMS analysis. They were rich in theanins, terpenes, which give it an astringent umami flavor. Contains several bioactive compounds contributing to antioxidant and anti-inflammatory property. This was the graph we obtained from GCMS of TS569. This is also a graphical representation of biochemical compounds. Then moving on to next slide. Result of P312. P312 had 73 detected compounds. Each showed higher concentration of floral and fruity volatiles like linalool and geraniol, giving it a sweeter taste than TS569. It was also richer in catechins and flavonoids linked to both flavor and health promoting effects. Surprisingly, this is a graph presented from P312. These are the list of compounds with area percentage greater than 5 in the GCMS analysis of both the varieties. Surprisingly, GCMS also detected environmental and industrial pollutants like dibutyl phthalate, likely from plastic containers, equipment, or packaging, hexachloroethane and pentachloroethane, industrial chemicals, which are industrial chemicals observed from polluted air, water, or soil. This underlines the importance of strict quality control, regular monitoring, and careful cultivation to ensure tea safety. This study revealed diverse biosynthetic pathway. This is even chain fatty acid biosynthesis pathway. Likewise, this is odd chain. This caffeine and oxalidinidiones. Terpene biosynthesis pathway, phenolic. And the next slide, comparative. This biosynthesis pathway explains the variety's specific differences in flavor and bioactive content. Comparative analysis. TS569 was rich in theanine and terpene, promoting umami taste. P312 rich in floral and fruity volatiles and catechins, sweeter and more aromatic flavor. Both varieties show distinct metabolic signatures directly affecting flavor perceptions. Now, the implications. This work has implication for tea breeding programs aiming for specific flavor profiles, quality control and contamination monitoring, understanding genetic control of secondary metabolite biosynthesis, developing sustainable farming practices to protect both quality and safety. And the conclusion conclusion, metabolomics allows us to understand the chemical and biosynthetic basis of tea flavor. TS569 and P312 differ significantly in metabolite composition, giving them unique taste and aroma profiles. Ensuring purity and safety is crucial before such teas reach consumers. Thank you for your these are the references, and we have come to the end. Thank you for your attention. Now I'd be happy to take your question. Thank you.
Thank you, Sakhi, for your wonderful presentation. Okay. Now I request Dr. Arup Sarkar. Arup Sarkar. Okay, I think he is not present. Now, next we go to the next Dr. Sumon Mollik. Dr. Sumon Mollik. Sumon Mollik, he is not available. Joita Sarkar, North Bengal University research scholar. Joita Sarkar, he is not available now. Now, next I request Dr. Kirti Ranjan Sahu. Hello.
Yes.
Yes,
I am.
Please,
present.
Please, sir, start your presentation. Okay.
Okay.
Now, Dr. Kirti Ranjan Sahu, faculty of physics department. Okay. Please start your.
Please remove the previous slide. The slides are now showing in the screen.
Please share your screen, okay, sir?
Okay.
Please share your screen.
Now, enter screen. Share. Enter.
Yes, sir. Now it is visible. Okay, please start.
Just a minute.
Now it is visible, sir. Please start.
Okay. Oh, okay. Good afternoon to all. Thanks to the authority, the international seminar organizers to arrange this type of program and to help me to present my research work in online through the oral presentation. The induction and enhancement of the superconductivity in BaFe2 AS2 by argon ion implantation from electrical and magnetic measurement, presented by Dr. Kriti Ranjan Sahu, affiliated in Department of Physics, Bhattor College, Daton Autonomous, Paschim Midnapur, West Bengal. Normally, this is a well-known matter. Fe-based superconductor like Fe AS2, BaFe AS2 sample, we prepared, and it is a non-superconducting materials because the presence of magnetic materials Fe. If we dope a compound like cobalt, here we implanted in doped in cobalt, and it form a superconducting materials, and the transition temperature increased due to changing their doping concentration. And it is well known matter. And there are several techniques. Here, there are several techniques to change the TC in the superconducting materials. The first, doping concentration changing, second, pressure change in the sample, and third, the ion irradiation on the sample. Here we mainly focused on the TC change due to the argon ion irradiation on the sample. The argon ion irradiation experiment have been done IUAC New Delhi, and the TC change measurement has been done in Indore UGC DAE consortium. Now of all we telling some about the character. The after implanting the ion irradiation, uh, Eisner et al. In 2018 show that the out of 52 samples of 55 compound, there are maximum decreases the TC, and most of three compound changes showing their change increase the TC, and the reported literature there are maximum changes 0.5 Kelvin in the compound FeAC 0.5 T 0.5 Kelvin. And this change, the Okaji et al. In 2018 Nature publication show that this change due to only the radiation nano micro scale change the strength of the compound lattice structure come to the point in our compound. Uh, in our sample, CO doped BAP2H2 compound, we changes the huge change observed and measured the electrical resistivity and the magnetic susceptibility. The change near about 8 Kelvin, that is huge changes. And second interesting topics is the after irradiation, the initially we starting we telling that BA Fe2AS2 compound normally a non-superconducting materials, but after the irradiation it transforms a superconducting materials. But how? Explain this, we come to the point. The CO doped BaFe2 compound, when BaCaFe2 compound with after irradiation, it changes their TC Kelvin due to the ion irradiation. They are form a nanoscale bubble argon bubbles, and they generate pressure, and due to this pressure the changes their huge amount of TC. This type of experiment, this type of observation, this type of invention the first and also published in 2015 Physicacy, published by our group. And the second observation and the focus point in this presentation, that the non-superconducting materials showing a superconducting nature due to same reason. They are formed inside a argon, inert gas argon bubbles, and they generate a pressure, change their structure, and they showing a superconducting materials. And um, uh, this is the our final work, presenting work, and acknowledge to supporting our our teamwork. Thanks to the Dr. Udayande, retired professor HBNI, and Dr. Thomas Wolf, Institute of Quantum Materials and Technology, German, and Dr. Ashish Kumar Mishra, UGC DAE Consortium Indore, Dr. Alok Banerjee, UGC DAE Consortium for Scientific Research Indore, Dr. V. Ganesan, UGC DAE Consortium for Scientific Research Indore and Medicap University, Dr. Ditto Sandal, HBNI Professor, VECC Kolkata, Dr. K.K. Chakraborty, Professor HBNI, and retired scientist, Bhabha Atomic Research Center, and Low Energy Ion Beam Facility, IUAC Delhi, and Low Temperature Unit, UGC DAE Consortium for Scientific Research Indore. And thanks to all for listening this type our lecture, and the Darjeeling Government College and the conference organizers. Thank you all.
Thank you, sir, for your presentation. Now I request Dr. Taposh Kumar Adildar, Chemistry, Department of Chemistry, Jhargram Raj College, for his presentation. Dr. Taposh Kumar Adildar, please start your.
Okay, thank you, sir.
Please share your.
Yes, I'm trying to share.
Please start, sir. Taposh sir, now you are visible. Please start your lecture. Okay. Taposh sir, please on your mic. Okay, I think it is in off mode. Taposh sir, you are not audible. Please. Okay, now. Okay, thank you.
So today, title of my talk is high throughput crystal engineering based synthesis of supramolecular gels, blue emitting fluorescent gold clusters synthesized and stabilized on gel bed. Before I am going to enter my work, briefly I share with you the concept of crystal engineering. The crystal engineering, that concept was developed by Professor Gautam Desiraju. According to him, the crystal engineering is the understanding of intermolecular interactions in the context of crystal packing and in the utilization of such understanding in the designing of new solids with desired physical and chemical properties. In crystal engineering, there is another concept, that is supramolecular synthesis. So supramolecular synthems are structural units within the supermolecule which can be formed and or assembled by known or conceivable synthetic operations involving supramolecular interactions. Now with this, I can compare the supramolecular chemistry with the molecular chemistry. In molecular chemistry, if our building block is atom, in supramolecular chemistry, our building block is molecule. In molecular chemistry, the atoms are linked together by covalent bonds. In supramolecular chemistry, the molecules are engaged among themselves with intermolecular bonds.